In today's set of lectures, we're going to talk about the mechanisms that are used in glycolysis. Now, I know the core metabolic pathways in glycolysis have been used, you've seen them in other classes and you know the structures, but the goal of this set of lectures is to describe the chemical underpinnings of those reactions and the logic that that's used in biochemistry. And as we're going to see in this set of slides, there's a recurring theme of types of reactions that we see throughout glycolysis. So as a brief reminder, so glycolysis is one part of central carbon metabolism, and it's involved with converting glucose into pyruvate lactate, which are then ultimately either used for energy or go into central carbon metabolism for making other building blocks of other molecules, or going into the TCA cycle and oxidative phosphorylation for energy, or they can be siphoned out and used for building new amino acids. Just a reminder that glycolysis is one part of this overall view. Now, some of the core lessons that you'll need to know for today are, one is that every one of these reactions that we're going to learn about happens for a reason. Like, these reactions, they're not random, and there's sound logic behind why each of the reactions occurs the way they do. So something that's really helpful in working through all these biochemical reactions, there are basically three central rules that allow you to predict most of the reactions that we're going to talk about. The first one is that acid-base reactions happen quickly. The second is that bonds that are beta to carbonyl are labile, and that means they break easily. And the third thing is that resonance explains an awful lot of the reactivity that we're going to talk about. So because resonance is an answer that you're going to hear a lot. So the mechanisms we're going to talk about are keto-enol tautomerization, acid-base reactions, additional elimination, aldol, condensation and cleavage, and shift-based reactions. So let's jump right into it. The first step of glycolysis is activating glucose to make this organophosphate that traps the glucose inside the cell and allows it to commit it to central carbon metabolism. So this step happens by hexokinase or glucokinase, depending on which cell you're in, involves adding a phosphate that comes from ATP onto this terminal carbon-6 hydroxyl of glucose. So the way this occurs is there's a nucleophilic attack of the hydroxyl onto this phosphate, which ultimately results in the loss of an ADP and the formation of this molecule. Now, if you are familiar with your organic chemistry, you'll see that I've skipped a couple of steps in how we draw this. So normally, if we were to draw this out via the full addition-elimination mechanism that's shown in organic chemistry, you would show the nucleophilic attack of the hydroxyl on this phosphate, which results in this intermediate structure here, where the ADP and the oxygen are still bound. So this structure then progresses by electrons resonating down from the oxygen, resulting in the loss of an ADP here, and ultimately a base abstracting the proton to deprotonate this hydroxyl. So these are the individual steps as drawn in a more familiar mechanism via organic chemistry, but we can actually simplify these into a one-step drawing here for biochemistry. Now, the reason for this, as we got to remember, is that these reactions are all happening in water at pH 7, and as a result, we can make some simplifications. In water at pH 7, there's always going to be water around that's available to deprotonate it, functional groups like this, and there are always be protons available that can enable these reactions. So one important difference between organic chemistry and biochemistry mechanisms is it has a readily available source of acids and bases to enable these reactions to progress, and they happen very fast. So in fact, so fast that you don't even really need to consider them in the reaction mechanisms. So let's progress along here to some other ones. So phosphoglucosamines, as you recall, the logic here for this is that we ultimately need to take this activated molecule glucose and make this molecule down here, fructose 6-phosphate, or ultimately fructose 1,6-bisphosphate, that will allow us to cleave across this bond right here And the logic behind that is it will create a symmetrical molecule that allows us to go follow a single path through glycolysis in the payoff steps and lower steps of glycolysis. So this reaction progresses here through the same set of steps that we have seen before in organic chemistry for ring opening. Quickly, the base abstracts a proton from this carbon 1 hydroxyl. Those electrons then resonate here, and that breaks this this lactam bond here, and that forms this aldehyde. Okay, this open chain structure. This is the same ring opening reaction you learned in your organic chemistry class. In the second step, a base abstracts a proton here from carbon 2. That ultimately results in this enol intermediate here. A base then abstracts this this proton again from carbon 2, and then or from the hydroxyl and carbon 2, and that ultimately results here in this ketyl form of this molecule. Then a ring closing reaction occurs where these hydroxyls out here on carbon 5 make a nucleophilic attack on the carbonyl, allowing the structure to close and then forming this this 5-member ring, fructose 6-phosphate. Now, as a quick reminder here, in biochemistry, we have some other shorthands. I'm just going to move my screen here so you can see that these phosphates can be drawn here in shorthand. And so this circle P that you'll see me using here, this is a shorthand for this notation right here. Okay. So the next step in glycolysis is that we need to ultimately make this fructose 1,6-bisphosphate. Again, the reason for that is that when this molecule is broken along this axis right here, this allows you to have 2 symmetric molecules ultimately that can go through glycolysis. That way you don't have to duplicate enzymes for 2 different sets of substrates. So this reaction progresses just like the hexakinase reaction we saw a moment ago, where you have a nucleophilic attack on the phosphate, and that ultimately results in the loss of this ADP. Now, again, I'm simplifying here our additional elimination step here into one step, but if you go back and review the hexakinase reaction, you can see that there are multiple intermediates that I'm simplifying into a single step. It's okay to draw this in biochemistry in this simplified form. You don't need to draw each of the individual steps, and this is convention for the field. Now, aldolase is ultimately the reaction that allows you to get these symmetric molecules, and it's going to progress via a very similar mechanism to the one we saw a moment ago with the ring opening reaction. This happens with the base abstracting the proton off the hydroxyl, just like we saw a moment ago, and that forming this ketyl form. Then in this particular case, we have a circumstance where you have a bond that's, this one is called alpha, this guy's beta, so the one that's 2 away from the carbonyl we refer to as beta. So this here would be the beta carbonyl, and this guy is labile. The reason for that is because of resonance. We'll see here in a minute. This base can extract this proton, and when those electrons resonate down to this position, the carbon doesn't have any extra space for electrons there. Otherwise, it forms this Texas carbon. So it's got to kick these electrons off, and it can do that because of resonance. These electrons can resonate out to the proton. This ultimately forms this aldehyde here and this enol form here. And this is called an aldol cleavage. Like, this is one of those classic reaction mechanisms I was telling you about before. Now, we can tautomerize this form from the keto form, from the enol form to the keto form by abstracting this base, or using a base to abstract this proton, forming this carbonyl again, and then reforming this molecule. Now, you should find this to be familiar. This guy here is dihydroxyacetone phosphate, and this guy here is glyceraldehyde 3-phosphate. So the actual mechanism that occurs inside the enzyme is actually a little more complicated, although the mechanism can be thought of entirely through the lens here of this classic Aldol condensation cleavage reaction. It's the actual chemistry that happens in the enzyme is a little bit more complicated. It does start with the base subtracting the hydroxyl proton again here, opening the ring. But one of the differences is it progresses through a shift base intermediate, in which case a catalytic lysine serves as the nucleophile that attacks this carbonyl. And in this case, then after it's formed this tetrahedral intermediate, the nitrogen has an extra set of electrons that can form another double bond here. That results in the loss of the water and this shift base, which is the intermediate. Now, this shift base acts kind of like a pseudo carbonyl, wherein you see again here we have an alpha and a beta positions here to this functional group, and you can see that if you were to abstract a proton from this position, thus kicking these electrons off, these electrons have somewhere to go. They can go here, and this can resonate back out. So this acts kind of like a pseudo-carbonyl, but the difference is that this substrate is attached to the enzyme during the reaction. And then it releases this guy here, glyceraldehyde 3-phosphate. Now the enzyme has to regenerate its catalytic glycine, and it does that by sort of doing the reverse reaction, where these lone pair electrons here on the nitrogen then resonate back out onto the carbon structure, forming this shift base again, and then that's susceptible to a nucleophilic attack by the water onto this position, then forming this, again, familiar intermediate, where the base abstracts the proton and ultimately regenerates your dihydroxyacetone phosphate. Pretty cool. Last step. Now this one's neat, because this is the magic that allows us to have just one set of enzymes in the payoff phase of glycolysis. And it converts this dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So we have just one common set of molecules going to the second half. The way this isomerization works is that a base subtracts the proton that ultimately goes into this. Again, this is a keto-enol tautomerization. And then from this enol intermediate, you can have base subtracting this proton off the hydroxyl again, and then forming an aldehyde. So one simple conversion here gets us back to a molecule that can be used for the rest of glycolysis. Now, this is one of probably the most important reaction in glycolysis. This is the one that allows us to have energy payoff for these reactions. This is glyceraldehyde 3-phosphate dehydrogenase, or GAPDH. This is the one that engages in substrate level phosphorylation. And let's talk a little bit about how that works. So in the active side of the enzyme, you have this thiol from cysteine that acts as a nucleophile that adds here to this aldehyde, glyceraldehyde 3-phosphate. When it does, it goes through this intermediate. Now it should look pretty familiar from what we've seen here before. But the thing that's unusual about this particular reaction mechanism is that when the base subtracts a proton, what leaves from here isn't the sulfhydro or the organic group, it's actually a hydride. Now it's important to keep in mind this is not a proton that leaves, because the electrons go with it. So when you have this new structure that's formed after it, it's lost electrons. So oxidation is this reaction is an oxidation reaction. This guy gets oxidized into here, and then of course the NAD is reduced because it's gained electrons. So the important feature here is that this is a hydride moving and not a proton moving, right? So then in the next step, the enzyme enables a nucleophilic attack of this phosphate onto this carbonyl that ultimately moves to that tetrahedral intermediate that we showed you before. And I'm just simplifying here to a single step where we regenerate our sulfhydro group, and you ultimately have glyceraldehyde 1,3-bisphosphate. So that's how that reaction works. In the next step, you have the first one of our payoff steps here in glycolysis. This is the phosphoglycerate kinase reaction. It says this reaction occurs as a nucleophilic attack of the phosphate here onto the phosphate, the organophosphate here in position 1, glyceraldehyde 1,3-bisphosphate, and or 1,3-bisphosphoglycerate. And this is an additional elimination reaction, just like the ones we've seen before. I'm simplifying into one step. That gives us ATP as a leaving group, and this gives us this glyceraldehyde 3-phosphate, or I'm sorry, 3-phosphoglycerate as an end product. So then in the next step, we have phosphoglycerate mutase, which is an enzyme that takes the phosphate from one position and puts it into another. Now, the reason we call this a mutase enzyme and not an isomerase enzyme is that the phosphate actually has to come off the molecule and be re-added to a new position. And the way this actually works in the enzyme is actually it sits on this transporter histidine. And so the way this reaction works is there's already a phosphohistidine that's sitting there in the active site. There's a nucleophilic attack on the 3-phosphoglycerate onto this phosphate that results in the generation of this histidine here, which is then available to engage in chemistry at the this is the carbon 2 position here. And in that way, you can regenerate your catalytic histidine and then have this 2-phosphoglycerate instead of at the 3 position. I'm sorry, that was at the 3 position, 1, 2 to 3. Yeah, moving it from the 3 to the 2 position to 2-phosphoglycerate. Okay. And then in this final step, we have the enolase reaction, which gets us into this very high energy organophosphate, phosphoenolpyruvate, which ultimately enables our final payoff step. To get there, a base abstracts a proton here from carbon 2, and that makes this enol form here, which is stabilized by magnesium in the active site. Ultimately, the oxygen electrons resonate down, forming this phosphoenolpyruvate, which is a very unhappy molecule. It's both sterically hindered and planar around this position, and very happy to give away some free energy here in the form of phosphate. And it does that in this final step, pyruvate kinase, where the phosphate again acts as a nucleophile attack on the phosphate, which ultimately goes through an additional elimination step to this pyruvate. So the final notes on this are that that each of these reactions are occurring through a steps that should look familiar, and progressing through a series of reactions that look like repeating themes. In the final step I want to discuss is what do you do with that pyruvate? Well, this NAD that we generated from glycolysis, or I'm sorry, the NADH that we generated through glycolysis is critical for the progression of the pathway. In order for the pathway to continue to function, we have to regenerate the oxidized nucleotide. The way that works is if you don't have an electron transport chain, the enzyme can undergo a reaction where the nitrogen electrons resonate out, ultimately allowing a hydride to leave the NAD molecule, attack the lactate and reduce it, or I'm sorry, the pyruvate, and reduce it into the lactate. So that gives you a oxidized NAD and a reduced lactate as an overall product. Okay, so this was a brief overview of reaction mechanisms. Today we covered the keto-enol isomerization, acid-base reactions. We talked about additional elimination, Aldol cleavage, and shift base. So these are things that you're going to see are recurring themes. And if you learn these guys well, you'll be able to predict reaction mechanisms throughout biochemistry using the principles we talked about today.